discussed once at ECVP, and I think then afterwards. So I'm really looking forward to hearing more about this, and welcome everyone who joined. Yeah, looking forward to hearing it.
Yeah, thanks for the invitation. It's great to talk to you. I'm sorry for all this mess. Like I said, I have many slides, so if you can look at my camera and I will do like some kind of sign or something, because I have really many slides and sometimes I go fast. So yeah, I introduced a little bit the talk, so we can start with the next slide, which is the motivation for this project. So I wrote there, ultimately we want to understand human visual attention. Definitely there are more important questions for humanity, but if we want to understand vision in humans, I think this is a pretty interesting question. Next slide. And for many years, this has been studied using computational models of visual salience.
Hold on, Jeff, you need to get rid of this. I think somehow you're interrupting. This is weird. Okay, I have no more control of my own screen. This is kind of crazy. Yeah. Okay, right. Okay, here we go.
Works, yeah. So like I said, there are recently, in the last few years, we already have very accurate models of computational visual salience. Next slide. And what this means is that given one image, next slide, we can have a global salience map of these images. And I have a few more slides showing more examples. You can go through them. This would be another example. But the question we wanted to answer here is, okay, what if several images compete for visual attention, like in this example, if we have two images side by side, like here? And we call this global visual salience because we actually want to understand what images are more likely to attract our attention. We can go to the next slide, which is the outline. And here I have many slides. So I always have this spoiler alert. I like to say the main findings at the beginning, and then I will try to convince you through it. So I will talk about the data collection first, very briefly. Yeah, thanks. Then about the computational model that we use to model visual salience. And then various results. And I have 5 main results here. You can go by one by one. So the first thing is that it is possible to measure global salience of natural images. Global salience of images cannot be explained by their local salience properties. We also found the strong spatial bias towards the left, which had been shown before. So this is good news to confirm this trend. D is that we didn't find any strong effects of task and familiarity, at least in the way we studied this. And finally, images with larger global salience were also explored for a longer time. And this is something that we presented in a paper that was accepted finally this year at the Journal of Vision. That's in the next slide. And I will finish the presentation with some words, some slides about what we are doing right now to follow up with this project. It's about the temporal dynamics of the trials. Yeah, thanks. So I will start with, this is one slide for the data collection. This is an eye tracking experiment. This little video here shows, it's not exactly what happens in the trials, but it is to show, well, it stopped anyway, but essentially we show these 2 images to participants, and they have to focus on the center. And then for these results, what we did was to analyze the location of the first fixation, so either left or right. And we had 200 images that could be categorized into 6 classes, 49 participants. And if you look on the bottom part of the slide, what we did to account for task and familiarity was to randomize if we present one image new and one image old, meaning it has been shown before by the participant during the experiment or not. And also we introduce some small, very light task, which was to select which was the new image or the old image after the trial. That was all. So in the next slide, I have the information about the computational model.
It's not letting me, hold on.
Almost there. Yeah, so we modeled global salience with a machine learning algorithm called logistic regression. Maybe it's familiar for most of you. And I will present a little bit the main details of this, because I think it is interesting to to see how it is possible to very easily model different metrics with such a classifier, which is very common in machine learning, but I feel it's not so common in cognitive sciences. So the goal here was to predict the location of the first fixation. Like I said, left or right given. So the input data is the two images that are presented to the participants. So with logistic regression, this is just very quickly the information about this. Exactly, we have the idea is to model the data X with a function H, which is a logistic function. And in the next slide, there is the plot of the logistic function. Yeah, probably most of you are familiar with this. And with this, in the next slide, I have the probability, the posterior probability of the labels, which is the actual location. And yeah, this is just a logistic regression 101. And the way this can be implemented is through a design matrix like this matrix X, which has everything that's in the data. In this case, the images that were presented in all the trials. And in the next slide, I have that's the Y. Go to the next, please. We have the specific case for this. Logistic regression can be used for with very complex input data, the features, and in this case we always have only two images. So we have this design matrix, and then it becomes much simpler. In the next slide, hopefully. Yeah. So for one trial, we have this is just one trial, and because we have 200 images, that's one row of the whole matrix. We have 200 images, so we have 200 entries in this vector. And next, exactly. So this is you can go a little bit faster. So here for a second. So in the end, all the entries, all the entries in the vector will be 0 except those entries that correspond to the images that are presented in that trial I. Okay, and the image on the left will be 1, and the image on the right will be -1. Thanks. Next. Yeah. So for this case where we have pairwise comparisons, this original logistic function becomes this, which is which is a simpler version because we only consider the elements U and V, the image on the left and the image on the right. Next. And the design matrix, this is the original, becomes a very sparse matrix with which is mostly zeros. I think that happens in the next slide. It's mostly zeros, and it will have the 1 or -1s in the corresponding elements for the images. What we did here, and you are seeing different additional columns, was to introduce the new factors for the factors we wanted to study, and these are task, familiarity, and the subject, the participant, which was performing each trial. Right, and this allows us to model these different factors and take them into account for the modeling of global salience. And in the next slide, there's the results of training this model. So we train it with the train data, and then we test it with the test data, of course, as is common in machine learning experiments. And what we show here, this is not the main results of this project. This is, you can see that the model is quite accurate. In general, this is only to say that because the model is accurate, then we now can use the weights, the parameters of the model to do more stuff with that. So this is kind of a proof of a concept to say, okay, now the model works. It is able to predict what happens in the experiment, and now we will use the parameters as the proxy for global visual salience. Yeah, thanks. So I'm already here to present the results in this slide. So first, what I would say is the general main result of the project. Like I said, there will be one coefficient, one parameter, one weight, call it as you prefer, one coefficient per image. I said there are 200 images in the experiment. So the higher it is, the more likely it is to attract the first fixations. So if we plot all the images sorted by their global visual salience, by their coefficient, this would be what we get. The top left image is the image with the highest global salience, and it goes from left to right and top to bottom. And you can see that on the bottom right, there are 3 images with which are pink noise that were there in the set for sanity checks, so they receive the lowest score, so it kind of makes sense. And then what we can see here as well is that images of faces and humans received in very consistently the highest scores, highest global scores, global salience scores. And then we have urban, indoor, natural images, which are more or less in between. Yeah, so the conclusion of this is that images with faces and humans are more likely to attract the first fixation. Yeah, thanks. This is interesting, but we wanted to make sure that this is meaningful in the sense that it is something that is not just explained by the local properties of the images. For example, you might be wondering, okay, but all the faces have a very special particular structure, and maybe the low-level properties of the images are for some reason more attractive. So for testing this, we use two computational salience models, GBVS, classic from computational visual salience, and DeepGaze2, which was at the moment the state-of-the-art for modeling visual salience. And this DeepGaze2 uses high-level descriptors from trained with a neural network on object recognition datasets. And for comparing the global versus the local salience, we performed two types of analysis. One was the predictivity, to test the predictivity of salience maps, okay, the local salience maps, to predict the location of the first fixation in our experiments. And the second type of analysis was comparing the predictivity of the salience maps with our global salience scores derived with the logistic regression models. Okay. Meike, by the way, how much time do I have for the presentation?
No limits. People can leave if they have to go. Otherwise, everyone is your captive audience.
All right. Yeah, so for the first type of analysis, the predictivity of salience maps for the location of the first fixation. Can you go to the next slide, please? For this, we use the Kullback-Leibler divergence to compare a map of first fixations. So we get the first fixation in our maps, in our images, sorry, and we can compute the salience maps. And we can compare this, which are essentially a distribution of fixations, to see how close they are. And it turns out that they are not very related. So the conclusion of that is that, well, the first fixation maps, sorry, the salience maps cannot predict the first fixation maps derived from our experiments. In this slide, I'm showing the results for GBVS, and in the next slide is the results of DeepGaze2, which in DeepGaze2 is a little bit better, but it's still not predictive.
Just 1 second, because there are people waiting in the lobby, and I need to somehow get back into controlling Microsoft Teams, which I can't. All at the same time.
I think I have to stop control.
Okay. Hold on. Do I need to admit someone? Okay, apparently not anymore.
Oh, I admitted them. That's why my cursor went on the screen.
Right. Okay.
Sorry.
No problem. Okay, I'm just going to try this again.
Yeah.
Okay.
Yeah.
Are we at the right slide, Alex?
Yeah, you can go to the next one, which is DeepGaze2. So here, what you can see, maybe this is a little bit strange, or I went through it very fast, but what you can see here is on the right, you see the global salience map, the local salience map computed with DeepGaze. So that's the heat map. And then you see the green dots, which are the location of our first fixations, so the first fixations of our participants in our experiment in the images. And when the Kullback-Leibler divergence is minimum, that means that these two maps are closest. You get that image, and yeah, in that image is kind of okay, but then in most of the images, the maps don't really correspond to each other. So yeah, so local salience maps don't match the first fixation maps. Yeah, so this is we can go to the next slide. This is to compare local salience versus global salience. We did other experiments. Yeah, we can go a little bit faster here. The details are a little bit maybe convoluted here, but in general, what we find is that there is no predictivity of the salience maps, the computational salience maps, with respect to the global salience. So here we are plotting the correlation. You can see with GBVS, which is the plot on the left, the correlation is very, very low. With deep gaze, it's a little bit better, a little bit higher, I should say. But still, it's still kind of insufficient to account for the location of the first fixation in our trials. And I have a couple more plots here, but I think I'll go a little bit fast over this. Yeah, this will take me a while to explain. So if we can stop here for a second. So what we are doing here is there are many dots in the scatter plot because each dot corresponds to one single trial when a participant sees two images side by side, and we are comparing the salience mass on the left versus the salience mass on the right, and comparing this difference, this absolute difference, with respect to the global salience. Okay, and still this is not correlated, so we couldn't find a way to map properties of the local salience maps and our global visual salience scores. And this is with GBVS. The next slide is with deep gaze 2. A little bit better again, but not strong enough. And we were surprised by this. We actually wanted to find a way to explain our visual salience with the local salience properties, but we honestly couldn't find it. Yeah, and I think we can go to the next. This is in this slide I'm presenting the results of the lateral bias, which is very consistent. So 63% of the first fixations, landed on the left side. The 4 blocks you are seeing here correspond to different blocks in which the experiment was divided. And in all of them, we found this consistent bias towards the left. And in any case, that was something. So we see this in general, but different participants have very different biases. So some of them have a very large bias towards the left, and some are even biased towards the right. And this is reflected by the standard deviation plotted in this around the means. And we did a little bit more to compute this. So if you remember from the slides on the modeling, we have coefficients that account for the bias of each participant. And again, as a sanity check, we can see that the percentage of first fixations on the right image, which is the x-axis, is very highly correlated with the coefficients learned by the model. And this, I will use this to highlight the validity of modeling behavioral aspects with this kind of machine learning models, provided the modeling is done in a meaningful way. Yeah. So here I'm presenting the results of task and familiarity. Just as a little reminder, the familiarity refers to the fact that in some trials, one of the images had been seen before in the experiment and the other image not. So again, we included coefficients for this in the logistic regression model to see if it's possible, if they contribute to the prediction. And in the next slide, I should have a plot that demonstrates that this is not the case. So we didn't find any strong effect of neither task nor familiarity. The task was simply to select the new image after the trial or the old image after the trial, depending on the block. And it was essentially, it didn't play any role. And finally, for this part of the presentation, we also computed some measures regarding the dwell time, so the exploration time during the whole trial for each of the images. So how much time the participants spent on the left image, how much time they spent on the right image. And we found that they have they don't explain the laterality. So this dwell time is not related to the laterality, to the bias towards the left or the right. But in the next line, we found that, or I don't know if I'm sure of that. Yeah, with task and familiarity, it is also not related. Yeah, sorry, with task is not related, but no, sorry, I need to again.
Sorry, do I need to go back?
Yeah, to the first one. Yeah.
Effect.
So
To
we
the
found
first
that
one, this one.
participants, yeah, the next one with the blue and with the
Okay.
purple and green. Yeah, here. So we did find a small effect and not very strong, in which participants did explore longer the new images if the task was to select the new image, which kind of makes sense. So they have to find, okay, what is the new image? And if it indeed was the new image, there was a new image, then they spend more time on that one. Yeah, this this kind of makes sense. And finally, in the last slide, we show that dwell time and global salience scores were moderately correlated. So this means that those images that received higher global salience scores by the logistic regression model also were explored for a longer time. Yeah, so to summarize this part, we performed this eye tracking experiment with computed visual stimuli, one image on the left, one image on the right. We also studied the effects of task, familiarity, and lateral bias by including factors, coefficients, extra coefficients in a logistic regression model. And we trained the logistic regression model with all the behavioral data, with the decisions, either left or right, the visual decisions, left or right. And the model was accurate, and this allowed us to use the coefficients as proxy for global salience scores. And we found that the images, natural images, are actually different in regards to their general global salience, but this global salience is not related or cannot be explained by the local salience properties. So to conclude all this, it is possible to measure the global salience of natural images, not related to the local salience, strong spatial bias towards the left, task and familiarity play a small role. Images with large global saliency are explored longer time, and I will also comment here that this I didn't include in the slide that images that have like faces or human figures in the image are the ones that are fixated first in the trial. And yeah, if you want to read more about this, there's the archive, the SciArchive paper and the Journal of Vision version, and all the code and data is on this GitHub repository. Also, the data is on OSF and so on. So if you are curious and interested or want to follow up or want to see if this reproduces, you can go to the GitHub repository, and hopefully everything is clear there. So I'm going to finish the presentation with a few slides about what we are doing right now, and this is work that has been led by Carla Matic, who is a PhD candidate at the Max Planck School of Cognition. I think she's in the audience right now. So here, what we are doing is studying the temporal dynamics of the decision making, the visual decision making. So what I have presented so far was all results where the location of the first fixation. If the image on the left, or the image on the left, or the image on the right was fixated. Can I have the next slide? Yeah, thanks. So if you remember, well, I don't know if I mentioned this actually, so sorry, but the trials were 3 seconds long. So we have more than just the first fixation. So some questions we were interested in answering was, how long does it take to decide where to look first, left or right? And what is the exploitation trade-off when we are faced with this competing stimuli presented side by side? And in the next slide, I have that this is related to the saccadic, or we can study this by the saccadic reaction time for the first question and the spatiotemporal dynamics to explore the exploration-exploitation trade-off. So the first result is that if we compute the average of the saccadic reaction time for each image, we find a very strong correlation with respect to with the global salience scores, which is interesting. So you can see that on average, images that are more salient, globally salient, are fixated faster. But this is not the whole story. This is, of course, it's the grand average. It's an oversimplification. If we look at every single trial, we see that things are subtle as well. So we went a little bit further to study what is actually happening with the location of the fixation during the trial and the time it takes to make the first visual decisions, whether to look to the left or to the right, and how this relates to the salience scores that we had computed in the first part of the project. Next slide, please. And yeah, so here what we are showing is we're dividing. So we want to consider the visual salience score, the global visual salience score of the two images. Okay, we get it from the previous results, and with this we can study how the reaction time varies depending on the combination. Is the left image very salient and the right image not salient? And there are different combinations, and we can see that the first saccade is faster if the salience difference is larger. So that would be like an easy decision. Like, for example, we have an image with a face and noise, for example. So then in this case, the first saccade is faster, but that didn't explain everything. Because also the sum of the absolute salience in the two images. So if two images are very salient, then the decision will also be faster. And we have more in the next slides to illustrate this. So this is divided into two plots. On the left, we have the difference in salience, so the image, the fixated image minus the non-fixated image. And on the right, we have the sum of the two images. And in both cases, we see a strong correlation with the global visual salience. Okay, so it's a complex thing. And here, we wanted to analyze what happens in the whole trial, depending on this combination of salience. So, and this is to see what is the exploitation. So do we spend more time on the first image or do we switch to the other one? So here, what I'm showing in this slide is the case where the first image, the image that is fixated first, has high global salience, and then you see in green, orange, and blue the cases where the other image in the trial has low, medium, or high salience, global salience. When I say salience, but I mean global salience all the time. Okay, so you can see here that if a highly salient image is fixated first, participants stayed on it longer time if the other image has low salience. That's the curve in blue. But it is not the case if the other image has high salience. That's the green line. Okay, that's interesting. And in the next slides, we have what would be like an error. What happens if we look first at an image with low global salience? Which, yeah, that's strange, but of course it happens sometimes. So in this case, when the other image, the image that has not been fixated, has high global salience, you can see that's the green curve. Then we switch, or participants switch to the other images very quickly, and the effect is smaller the lower the salience of the other image is. And in the next slide, I have the plot for the case of the medium. The first fixated has a medium average global salience, and you can see that there is a trend from high to low. So, and yeah, and to say a few words about that's about the beginning of the trial, and you can see that as the time goes on, the two images are more or less similarly probable to be fixated or explored. And next slide. To conclude this, or to the main conclusion of this results, is that the global salience that we have derived in what I explained at the first part is able to explain to a large extent this exploration trade-off. If we stay on the image that we fixate first, or if we switch to the other one, and this is true at least for the first part of the trial. Yeah, so to conclude this part and the whole thing, because I'm finishing, we found that salient images are actually fixated faster on average, but this reaction time depends on both, at least both the difference between the two images and the absolute salience of the image. And the global visual salience balance, so this difference between the two, can explain the trade-off between exploiting the first image or exploring both. And with this, I conclude the presentation. thank you for your attention. And I have one more slide with I don't with the main plots of the presentation. And I'm happy to take questions if you have any.
Thank you. Thank you very much. Okay, I'm going to minimize the slides if I manage to, and maybe that will facilitate asking questions in case I have to point to something that any related questions come up with.
Thanks very much for for the multitasking of.
That was interesting. I definitely feel that it came at the detriment of the content to some degree, but it was also an interesting experience to try to anticipate someone else's slides and organization of thought. Okay.
Yeah, I could see that.
Yeah, sorry.
No, I mean, it was
All
good,
right.
like predicting the.
Okay, so any questions?
I'm happy to shoot first, actually, if nobody else is.
Go for
Hi.
it.
Thanks, Alex. This is a really interesting talk. I was wondering if you could go back in the slides to where you were showing those 3 graphs with the trends at the end. Yeah, 102. Yeah, 102. Oh, I guess Meike has to go back for you.
Yeah, I'm there.
Thank you, Meike. So I was wondering, in this case, there's a task context in which people are looking, let's say, at one or the other image, right?
Mm-hmm.
I'm wondering what the reaction times look like for the tasks, in the sense that if their task ends around the time that they stop exploiting, or sorry, stop exploring and start exploiting, right? If the reaction times tend to be around, let's say, that 1,000 to 1,500 millisecond mark, then that's sort of, you would expect their fixations to come back to chance because the task part is over. So I'm wondering to what extent their, let's say, reaction times for the task depend or are sort of correlated with this trend.
Yeah, that's a good question, because I think it comes from the fact that there are many experiments in which the task is actually, the task defines the reaction time. In our case, it's a little bit different. So we are calling reaction time to the time it takes us to look from the center to one of the images. And what I've discussed about task is always at the end of the trial, so after 3 seconds. And only in a few in 2 blocks of the experiment, so in half of the experiments. So after the participant has seen the 2 images for 3 seconds, and they always have a free viewing, then they are asked, okay, which of the 2 images was left? Which of the images was new or old?
Right.
And that's it. So, and we haven't analyzed this yet for this this new part of the spatiotemporal dynamics, but we found that there is no big effect of this task in which image we looked first.
Right. I mean, if you're only asking after the trial, it's hard for them to. It's hard for you to make a decision about, like, well, okay, they must have finished doing the task now.
Exactly. So the task, I mean, that was like an additional add-on to the project to say, okay, what if the participants have to do something after the trial? But in general, this, because it's a very light task, it has little effect on the decision, on the visual decision, at least the first fixation. But I appreciate your question in that we will have to look at this, at the effect of task for the dynamics. It might play a role. I'd be surprised, but yeah, we need to check that.
Can I jump in with a follow-up here?
Yes.
Absolutely.
Yeah, thanks. Yes, I think it's an interesting point, and what we might actually expect is that there's going to be more exploration in the case when there is no task, and more exploitation in the case when there is a task, because we have seen that people spend more time with a new image when they have the task to identify the new image. So in those cases, you probably would expect more exploitation than when there is completely free viewing and absolutely no task. So I think that's actually a very interesting hypothesis that I didn't really think of before.
That's kind of cool. Thanks.
Just checking if there was any questions in the chat, and I was just saying thank you. Peter. did you want to say that?
Yeah, I'm getting very confused with all the different things appearing on the screen here.
Sorry.
Sorry, it's just getting myself back from it. So I was thinking about, I don't know, 15 years ago, Simon Phillips' group had data that says you can saccade astonishingly quickly to the image that contains something animate. And the reaction times were something like 120, 130 milliseconds to go to the one that has an image, and 140 milliseconds to go to the one that's showing an animal. And the suspicion at the time was, yeah, there's got to be some low-level image properties, but the analysis at the time couldn't show it. Do you know, have you tried this? Is there a salience difference? Could you try it?
Yeah, this is actually the question we try to answer. So thanks for asking this. This relates to the set of experiments we compare it to the local salience properties. For us, when we see that, based on these experiments, well, when we see that we very quickly fixate or make the decision to fixate at faces or images, here we don't, or humans, sorry, here we don't have animals and a set of data, set of stimuli that actually tries to answer this. So animated versus inanimate, but we have that component for sure. So when we saw that, we wanted to find how could that be explained by local salience properties, and we tried many different things. We didn't find any way of doing that. The only indication of something that could explain it is kind of a trick, because we only found a little bit of correlation from local salience properties, but only from this model, deep gaze, which has been trained on object recognition. So somehow these salience maps do have information about the object categories. Okay. So in the end, it seems that we make this decision based on probably peripheral information during this very short time.
Okay, right, thanks.
Thanks.
I've got a question too. I'm not exactly sure as to how comparable your results are, but I remember seeing a talk from Stephen Schulte once at the University of Louvain when he was looking at the low-level image statistics and how they would affect perceptual decision making. If I recall correctly, he presented the images not in such a divided visual field manner. I'm not exactly sure, but I remember him talking about, you know, the cluttered and complex scenes with different types of values and Weibull fit and all these, like, how does that add up? Because I feel that his results suggested that low-level image statistics would account for a lot of the perceptual decision making. And how does that either add or contradict, or how is it dependent on the methods and paradigm that you use?
Yeah, I think there are some studies that have looked into this. I have the impression that most studies don't have this kind of paradigm where you have a gap of, you know, like a blank gap, and only in the periphery you have the stimuli. And this is our case. And here we didn't find effects of the low-level properties. In other studies where you do have visual information, rich visual information around the fixation points, then I think it makes more sense to find an effect of the image statistics. Mm-hmm. But here, like again, we tried and we didn't find a way to account for the decisions. And very consistent. It's strange because very consistently. So, for example, we have these images of faces in our set, which are very different to the others. And we could say, okay, yeah, the image statistics are different, so maybe that this is driving the first fixation. But the image statistics of the urban or natural landscape are very similar to these images we have in our data sets, which are essentially the same, but maybe like two people is very small in the scene. Mm-hmm. So, and they are very, very consistently fixated first. To me, that's strong evidence, strong for the kind of data we have. Of course, I won't claim that it extrapolates to anything, but for this kind of data, that's this strong, consistent behavior points towards this, that it's something of higher level that is already processed in the periphery.
Okay.
Out of interest, have you tested what happens with upside down faces?
No, we haven't.
Because image statistics are identical, so I don't actually know whether people do find them less quickly. I can't remember. I'm sure it's been tested, but I can't remember if that's true. But
yeah, according to the results that have been shown in like long time ago in literature, I would guess that they shouldn't be fixated first, like the upright faces. But we haven't done these experiments. But that's a good point. I'll take a note and see if we can study this. Thanks.
Chats, you want to go?
Yeah, sure. I was just waiting for him to take his note. So I'm wondering, because you've used a couple of models that deal with, let's say, the low level properties of these images from a sort of bottom up visual standpoint, and then I see the indication is that, like, well, the rest must be the more top down structural bits of the scene. And I'm wondering, so for example, I know Ode Oliva has like a GIST model for at least scene perception, which I think goes sort of one step beyond the low-level features, at least to explain like what the scene's layout looks like, and what parts of the layout are sort of best suited to helping people make decisions about category of scene, like whether it's an indoor or outdoor scene, that kind of thing. I wonder whether you've thought about picking the low-level models about any high-level models to see sort of what's the part in between that neither one of those things really explains, if any.
Yeah, it's kind of a open question. Yeah, sure. This has to be studied further, and I'm aware of these scene recognition models, and it would be interesting to use these features for studying this. I would actually take a different approach to follow up with this, which would be to collect more data, more eye tracking data with this kind of experimental setup. And because if we are interested in, okay, where do we look first, and what drives our first kind of fast fixation, then I would collect more data. It seems to be quite consistent if you have varied stimuli, and then I would train like machine learning models, like deep networks, specifically to perform this task.
Right.
That's essentially what we did, but instead of using just the behavioral information, so the decision left or right, to use the image as input. And if a deep neural network is able to predict the location of the fixation using the visual information, so the image itself, that would be the first step. If that works, then we could look at what are the features that the deep neural network uses to explain what happens here. This is the approach I would take.
Yeah, I like that as an approach, because then you're skirting the issues of, well, if I take a scene perception model and try to make it perceive faces, what's it going to do? Or equivalently, like for a face perception model, what's it going to do when I ask it to perceive a scene?
I think it will that approach would tell us something, but then we will be kind of stuck with wanting to know more and wanting to.
Oh yeah, absolutely.
That wouldn't give us the whole story. So that's why I would train a model specifically for this task, to see what are the features that are uniquely obtained for performing the task, and not for a kind of a related but not exactly the same.
Yeah, thanks. That's a good idea as an approach goes.
This is a side question, but I'm just wondering whether you have tried doing this sort of eye tracking at all with any of the webcam-based eye trackers.
This is we haven't yet, but that's in the plan. So now that I mentioned this kind of experiment with deep learning, we will need more data, and a webcam would be just perfect for this because so if we are interested in left or right, we don't really need to get very precise information of where exactly we look. And a webpan is perfectly fine, and that would allow us to collect a lot of data. And I recently talked to Graham Taylor at the University of Guelph in Canada. He's a machine learning scientist, but he was interested in exploring this, and we talked a little bit about this. So we're actually thinking of using web-based eye tracking. Web-based eye tracking systems to collect this kind of data. Be very cheap and efficient.
Yeah. The only thing I would be worried about in doing that, just from what you have said, it sounds like there are fairly prominent eccentricity effects. So like depending on how much visual information is how close to your fixation. So if you open this for something like online testing, you have to make sure that there's some let's say constraint on the image sizes that people can be viewing or the distances at which they can be viewing from.
Yeah, that's right. I'm glad you mentioned this, and I would ask the question back. So what would you put most attention on with this kind of, because I also have the intuition that, if this is something that has to do with the periphery, and it clearly seems to be the case, then that is very important. So how much periphery, how far from the fovea we are going to place the images, and how are we going to control for that? Yeah, that's an important question, and I don't have yet the good idea of how to control for that.
I mean, if I had to come up with a naive example, I would first take measures to make sure people can't use a phone or a tablet, say, just because your screen sizes will be too small.
Thank you.
and second, I would say if you give people a task where they're presumably like pressing a button on the keyboard, then you can more or less pretend that your arm is, or think about like what's the average size arm that a person can use, sit yourself about as far and as close as you would be comfortable doing it, and pick stimuli that in either case will be, let's say, 5 degrees eccentric at minimum from the that way you're at least not in paraphobia.
Yeah. It's still hard to. For me, what I find tricky now is, okay, how do we determine what is the right distance? Then the big data approach would be to collect a lot of information and collect it just at different distances, so different visual angles with this online experiment, and then like answer that question on the same go. But that would require a lot more data.
Wouldn't you be more concerned about the person maintaining a stable distance? Because I think that would be the trickiest part, right? Okay, yeah, you could have different sizes and different degrees of eccentricity, but if it's not maintained consistently across a session, I don't know. So we would find a simple, stupid way of just creating something like a cheap head fixture or something that people can measure. Yeah.
No, that's a good point. But I think if we design the experiments, in a way that, okay, now we found that this is why it's good to have this previous experiment. No, we see that this familiarity task don't matter, so we remove this from the experiment. Just very simple, 2 images, a lot of trials. Then each trial will be, to a large extent, independent from each other. So that would be the assumption. So from a given participant, it wouldn't really matter a lot if for different trials they move back and forth, as long as we have this information, which can be taken from the eye tracker on the webcam. And then we will simply have a lot of data, a lot of trials, and for each trial we have the distance. So we have the measure of the eccentricity at which the 2 images are placed. And with enough data, we could answer the question of how that affects the visual decision making.
Plus, as long as the images are sort of eccentric enough, there's a good chance that you won't get very large effects of viewing distance anymore, just because there isn't enough room for sensitivity to spatial frequency to drop off at that point, right? Like they're already more or less working with the minimum of their contrast sensitivity functions.
Yeah. So at some point, if they are far apart enough, then it would be like random.
Yeah.
I expect this, but we need to see, or we would have to find out what is the threshold for this kind of data, or this kind of decision, I should say.
I mean, the ideal situation would be, it's probably really hard to do it online, but if you have people, expressly using their webcams, you might be able to do something where you get, let's say, a measure of interocular distance from the person, and then based on how their interocular distance is changing, you decide how far apart you place the images so that they should be at roughly this person's current viewing distance for each trial. That might be one way to do it.
Tricky to do it online in this benefit, sure.
Yeah, I agree. You may have to have a thing where, kind of like in eye tracking studies where they make you maintain fixation for a second or something before the next trial. You do something where we need to pick up your interocular distance right now, so you have to sit tight for as long as it takes for the webcam to pick it up. And if it does, go to the next trial. If it really can't, you can press a button to go to the next trial regardless, and we'll discard it or something like that. That might be one way to do it. I don't know. I haven't tried this. I'm spitballing just to.
No, but that's good to hear you. Thanks. Yeah, I hope we get to do these experiments and we'll force ourselves to think about all this.
Yeah. Well, I mean, especially during COVID, it's a great time to be thinking about sort of alternative means for gathering the data that we actually want, as opposed to just the data that are available.
And also thinking about it with all these technical constraints that are really important and worth considering. So
yeah. Have you done?
It was, sorry.
I'm asking if you have done like web-based eye tracking experiments yourselves.
Eye tracking, no. Eye tracking, no. I haven't. Peter, are you guys running anything eye tracking?
Not actually tried it yet. No, I've got a student who wants to try it as a project, so I'll see what she does.
I can say I've used. Sorry, go ahead.
No, go.
Oh, I can say I've used, let's say, webcam-based eye tracking before, but not like at a random setup online. We had like a setup sort of desk to simulate kind of an office situation, and had people sitting at the same desk at roughly the same viewing distance. So we didn't have this weird extra constraint of we don't know how far from the computer your keyboard is going to be placed and how big your monitor is and all these other things. So
yeah, I think for many paradigms that's very important to control, but for this particular case it doesn't seem so critical unless I am missing something important.
No, not really.
We couldn't reproduce probably the last part. Like I think I would be more careful to try to analyze the whole trial, but if we just look at the first fixation, then it seems plausible.
Okay. Thank you very much, Alex. Okay. I'm going to go, I think. But cheers.
Okay.
Thanks
right.
for discussion. Thanks for inviting me. And
really cool talk.
Yeah.
That's a lot.
Thanks, Mike, for organizing.
Thank you. Thanks, Peter. See you soon.
Bye-bye.
So we'll send you the recording then. Thanks also, Carla, for joining. And yeah, thanks for presenting, Alex. We'll send you the recording, and then, or I guess now Jeff can take over and send it to you.